Welcome to the Proteomics in Proximity podcast, where your co-hosts Cindy Lawley and Sarantis Clamidis from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Cindy and Sarantis.
What if your chronological age only told part of your story? What if your brain, your lungs, your ovaries, what if they were all aging at different rates in your body? And that you could know that, you could find that out from a simple blood test. Today's guest, Tony Wyss-Coray from Stanford University, will talk about just that. He's one of the pioneers who's building organ aging clocks out of simple Olink protein measurements. Tony and his team at Rise are revealing remarkable insights into biological age, organ health, and even future disease risk years before symptoms. At Olink, of course, we're delighted to have our customers get more out of the data. So we're partnering with the team at Rise to ensure customers do just that. In this episode, we talk about young blood, vampires, immune cells, organ aging clocks, and so much more. I hope you enjoy it.
Hello, everyone. Welcome back to Proteomics in Proximity. We're very excited to have a guest we haven't had before here to talk about aging and some amazing innovations happening in aging. Sarantis, do you want to introduce our guest?
Thank you. Thank you, Cindy. Welcome, everybody. We are excited and honored to have with us a leader in the field of aging research, Professor Tony Wyss-Coray. Tony is a professor of neurology at the Stanford University School of Medicine, director of the Phil and Penny Knight Initiative for Brain Resilience, where he leads pioneering research on aging and age-related diseases. His work focuses on blood proteomics and circulating factors, and how they influence health span and lifespan, with a goal, of course, of developing interventions to help us live longer, healthier. Tony, it's a pleasure to have you with us. To start, we would love to hear a little bit more about your journey. What led you into aging research? And what continues to inspire your work every day in the lab? Thank you again.
And it's hot right now. This aging stuff is on fire. It's so exciting.
Yeah, thank you. Thank you so much for having me. When I started out in research, I started actually in immunology. I really had no interest in aging research, to be honest. But I got into neurodegenerative research and studied how the immune system affects the brain, and particularly in Alzheimer's disease. And I realized that age is really the key risk factor for this disease. And we did more than 20 years ago some very simple experiment where we asked patients for blood samples and asked, could we see changes in immune factors in the blood of these patients with the disease compared to those without? We saw some differences, but we realized that in the healthy controls, the changes were actually often bigger. So there were a lot of changes in the composition of proteins that we measured. And at the time, it was actually a filter array where we measured 120 proteins. And these changes in healthy people are often bigger than between disease and control. And that really got me into aging research.
Meaning that over time, the longitudinal aspect showed changes despite them not having any apparent disease, but that there were changes that happened longitudinally that were, because so much of the big population health projects are one sample, right? The baseline sample, like in the UK Biobank data, which went into the paper that we certainly want to talk about, the organ aging paper today. But those are baseline samples. And so this was really about longitudinal sampling, way ahead of its time, I'll say. 120 proteins, it's a lot at the time.
Yeah, it actually wasn't longitudinal. It was still cross-sectional. But what we saw is that people who were younger had different concentrations of proteins than those who were older. So this was cross-sectional, but it just showed that, age seems to be associated with changes in concentrations of proteins. And, now this makes total sense. And we know, of course, most molecules change in concentrations with age and in organisms, and certainly function changes with age. We look different as we get older. So the fact that we can capture this in the blood provided us with an opportunity to start asking, are these changes cause or effect? And we can talk a little bit about what that led to.
Yeah, definitely want to talk about causality for sure.
And I'm guessing that this is what drives you later on to Parabiosis experiments, right? Because it was a big, actually, discussions. They make a lot of impact in community also, these Parabiosis experiments.
Parabiosis. Yeah, let's explain that.
Yeah, so exactly. So Sarantis is actually absolutely right. So this was really the motivation for us to get into this model. And so basically what this is a model where you can pair animals of different composition or different age or different genetic backgrounds. And in this case, and it's called parabiosis, so it means living next to each other. But essentially, when you use this model, the circulation of two mice, in this case, is shared. And so we paired young with old mice, and we did this actually in collaboration with Tom Rando, who in a seminal paper with the Conboys showed that he can change the age of muscle stem cells. So he used an old mouse, which has very low stem cell activity in the muscle. He paired it with a young mouse and could show that this regenerated and rejuvenated the old muscle. And so he had this model, and he actually recruited me to Stanford. So we asked, what is the effect on the brain? Because we saw these changes in blood composition with age. And again, this allowed us to ask, is this cause or effect? Are the changes a reflection of the aging organism, or do they even influence it? Turns out they do both. And this is really the basis of now the diagnostic potential of this, and the implications for biology in general. But what we did is we basically used the same model that Tom had used and asked, is the brain of an old mouse affected by exposure to a young circulation, so young blood factors? And that was indeed the case. So we could rejuvenate the old brain. These mice had less inflammation, more stem cell activity. But then what we also showed for the first time, actually, this was Saul Vileda, who has now his own lab at UCSF. He showed that he can simply take blood from young animals and repeatedly infuse it into old. So he did this every 3 days for 3 weeks. He infused small amounts of just the liquid part of the blood, actually, not the cells, just the plasma, and could show that this reproduced pretty much the effects of this parabiosis model. And most importantly, in the improved function of these mice. So when mice get old, they get cognitively impaired. They have difficulty navigating a maze. And when they're exposed to young plasma, they can navigate it better and their function improves. And so that proved to us that there are factors in the blood that can not just reflect the aging of the organism, but they can actually change the age of an organism.
That's just amazing. I mean, from the way that I understand is like these factors, they target specific cells in brain, right? Because at the end, the brain seems that is the final actually hit on these factors. Do you know what type of cells are you target with these factors? Are you aware? I'm guessing most of them they are stem cells in neuron.
Yeah, that's a great question. So initially, you can imagine when we first reported these results, and also when Tom reported these results, people thought that's too good to be true. How could young blood just have these effects? That's folklore, right? People thought, give me young blood and I feel...
Dracula.
Exactly.
A little Dracula stuff.
But then with the advance of single-cell genomics, where we can take cells basically of every organ tissue in the body of a mouse, we can study how every cell is affected by these factors. And so because we saw these effects, I was really interested in harnessing the transcriptomic approaches and worked with Steve Quake, who was one of the pioneers in developing these technologies.
First genetic diagnosis made by Steve Quake on himself.
Yeah.
Fascinating.
Yeah, so we built these atlases we call tabula muris, where we profiled the cells of all major organs. And then we used this approach to ask in a very unbiased way, how are the cells in the parabiosis model affected? Which cells, as you asked, Sarantis, which cells are most affected globally throughout the organism? We find that stem cells are a major target, especially hematopoietic stem cells, but also hepatocytes, for example. And as you know, hepatocytes produce the majority of proteins, at least in quantity, in the blood, like albumin and coagulation factors, complement factors, and so forth. But what was really also exciting is that almost every cell responds to this heterochronicity. So whether you give an old animal young blood or a young animal old blood, which accelerates aging, most cells show transcriptional changes. You can then ask, what are key pathways? One of the key pathways are mitochondria, for example. There's a general reduction in inflammation from young plasma to old animals. And I think with respect to the brain, we don't know exactly how these factors get into the brain, but there is sort of a reduction in inflammation in the vasculature. So for that, factors don't even have to go into the brain. And that may be one prominent way to get benefits. But we also know now from other studies that Andrew Yang in my lab, he now is at the Gladstone Institutes, he simply labeled proteins in the plasma and injected them into mice and sees that they're actually broadly taken up into brain tissue, unexpectedly. People always think there's this barrier, nothing goes in. This is totally not true. So we're now following up on different cell types. Neurons take up proteins from the circulation, but we pursue a very specific type of microglia that are specialized in taking up proteins from the circulation. So I think there's a whole biology of how this may actually work. I think the key message was really that factors in the blood can modulate aging.
And so let's define hepatocytes, right? These are the functional cells in the liver. And as you said, they are incredibly important in producing proteins that show up in the blood. So I think that leads us down a path of proteomics that you've explored since then. Can you tell us about how you got to where we are today? And in particular, I'm pretty excited to talk about the organ aging work that your team published in Nature Medicine. So this was Plasma Proteomics links brain and immune system, your foundation in immunology, which is makes complete sense that the immune system is key to this, and health span and longevity. So maybe it's useful to define health span as opposed to lifespan, and then talk a little bit about the path that led you to work with the UK Biobank data and build the organ aging clocks that we we're excited to see leveraged today.
Right. Yeah. So maybe just the definition of health and lifespan. So health span is described as the time of your life where you're generally healthy and you don't have any major diseases. So now I think the field sort of describes that we live roughly 80 years of age. In sort of economically developed countries with good healthcare systems. And of those 80 years, roughly 50, 55 years were totally healthy, the average population, right? There's, of course, always people who get sick at different ages or different times of their life. But the 55 years would be the health span. And then we're getting more and more diseases as we get older. And some people call this sick span. But the goal really of efforts in this field of aging research is to extend the health span so that you live healthy until you die, ideally.
The sled ride to the bottom. Yeah, I just want it to...
Yeah, you fall asleep or you say, I'm tired, I think I've seen it. And, you're 80
and you're done.
90 years old.
Yeah.
And that would be it. I mean, that would be a nice way to die, I think, rather than suffering from multiple degenerative diseases. And I think the worst for me, but for many other people, would be to lose your cognitive abilities and don't even know, who your children are, who your loved ones are, things like that. Yeah.
Yeah, those big ones: cardiovascular disease, cancer, Alzheimer's disease, or neurodegenerative diseases.
Exactly.
Yeah, those are the biggest risk for getting those is living long enough to get those.
Yeah, that's true. Yeah. But maybe getting back to how did we get here? So having shown that the blood is really sort of able to affect the aging or even the physiological state of cells in the body. Of course, we wanted to know where do these factors come from. That was one of the motivations to do the single-cell transcriptomics and ask, well, which cells are potentially producing factors, but also which cells are targets of these factors. And so the question was then, more generally, if we look at the changes in the blood, can we ask where these factors come from? And does that tell us something about the state or the physiology of a given cell or organ? And that's really a very trivial question if you think about it. And we've pursued this for decades, in clinical chemistry. If you go to the doctor, they take a blood sample and they measure a number of different analytes. They measure lipids. They measure also a few proteins, very few actually. But one, very prominent, to come back to the liver protein that is measured are transaminases, liver transaminases. They tell you if the liver is damaged or not. So if they go up, that means your liver is not really functioning well. But we've used this mostly in medicine to record abnormality, to record pathology. So people go to the doctor when they feel sick, and then the doctor says, yeah, I can see your liver values are too high, or, there's something wrong with your heart, based again on a couple of proteins that are measured from the heart. We measure, of course, some lipids, cholesterol. That's a prognostic factor, but we have very few factors that really look in the future. So we asked, can we get information from the brain in the circulation? So are there proteins in the blood that might be derived from the brain? And what was really sort of a revelation for us, once we started to use these multiplex platforms, like the one you are developing or have been developing, Olink, what we see is that there's lots of proteins in the blood that are not made as secreted proteins. Remember, I said we started out by looking at immune factors in the blood because I thought, well, the immune system sort of communicates across the body. So we may find some cytokines and chemokines that tell us about the state of the organism. But it turns out you can literally measure almost every protein that the body produces in the blood if your assay is sensitive enough. And one of the proofs for that statement is that we can now measure A beta, or tau, the key pathological hallmarks of Alzheimer's disease, in the blood. If you told anybody 20, 30 years ago, they would have said that's not possible. That will be too noisy. We will not be able to pick this up because there's no relationship. But it will also not be measurable. And now we can measure transcription factors. We can measure kinases. Any protein seems to make it somehow into the blood that could then potentially tell us something about where does that protein come from. And if we know that, does it tell us something about the cell where it comes from or the organ that it comes from?
Yeah. So plasma, I've thought about this a lot, and I'm curious because I don't necessarily think of plasma as, or even the organ aging clock, which we'll talk about in this paper, as a reflection of the tissue state as much as a reflection of, secretion, leakage, turnover of cells, signaling between cells, maybe between organs. But maybe because of all of those activities and the way they change with age, then maybe plasma does give you tissue state.
Exactly. Yeah. I think, intuitively we think about the proteins in the blood, maybe like you just stated, right? That they're a reflection of some either secretion or damage. But it seems not to be the case, because in a young, healthy organism, you find any type of proteins, again, cytosolic proteins that were never meant to be secreted, synaptic proteins, and I think they're a reflection of cellular activity. And many of these proteins, or maybe the majority, certainly of intracellular proteins, are released through some vesicular mechanism, exosomes. Maybe there's others that we haven't really characterized or described well, but exosomes is, a key part of this. And that's part of a communication system that people start to discover and describe. So, immune cells, in particular, macrophage type cells, they release tons of these exosomes, and maybe it's a part of communication with the rest of the body. Cancer cells, of course, also. But it turns out every cell can release exosomes, and people that now describe markers in these exosomes that tell them the exosome comes from a neuron, or the exosome comes from a macrophage, or it comes from a liver cell,
or a tumor, or a cancer.
When we then apply the plasma proteomics and measure the concentration of proteins with your tool, we look at all the proteins that are present in the blood, whether they're in a vesicle or not. And so we really have all this information wherever it comes from. Some proteins are secreted. They're meant to be secreted as an endocrine communication, but and others are shed from the surface. And if a protein is shed, it may also have a physiological function. And yet others come through these, exosomal mechanisms and other parts. And of course, some come from damaged cells. But I think it's really a combination of anything that happens in the body. And I really state now in all my talks, the blood is an endophenotype of organ physiology.
That's great. That's amazing. Yeah, that's a good point. Tony, staying on this, and I want to follow up on the question now, having technologies that only democratize proteomics and you can identify much more proteomics and low abundant proteins. Now we are able also to predict, right? To have biomarkers with predictive power. You mentioned the p-tau, right? The p-tau is already for late stages. Already if you have a p-tau, you are sick. I think now what you bring in the field and how you are the pioneer in the field is like having predictive biomarkers at the level of organ. And that's really a super powerful for precision medicine.
Yeah.
How would you see this moving forward?
Yeah, I think it has the potential really to change the way we do medicine and apply sort of this concept of, blood pressure and cholesterol, which are predictive markers, to many more organs by simply looking at molecules that are derived from these organs. And if they change with age and physiology, and I really see the state of an organ reflected in the age-related changes. So in other words, if the function of an organ changes, it is because the molecular composition of the organ changes, and we can measure that in the blood. So in other words, we can measure how your liver changes with age, how your heart changes with age, how your brain changes with age. And if it shows an accelerated aging, it is more likely to develop what we call disease. Now, disease is a human construct, right? And Vadim Gladyshev, his analysis of the same data we analyzed, his conclusion is that all these diseases that we give specific names are a reflection of accelerated organ age. It's a very bold statement, but I think he might be right for many of them. Of course, if you have a pathogen that causes a disease, then that would be exempt. But these chronic diseases of dysfunction of an organ, like Alzheimer, they're a reflection maybe of the aging of that organ and the failure of that function. We give a name, we call it a disease. Now, we know, of course, there's many different diseases in the brain, and the brain age, for example, is not a good predictor of Parkinson's disease. But I think that's just a question of resolution. As we get more and more proteins that are specific to different brain regions, for example, we may pick up a signal from the substantia nigra or from specific neurons that degenerate in Parkinson's disease. And that will give us information about Parkinson's disease. And in fact, we just showed this recently with measures that estimate the age of cell types, not just organs, based on changes with age. And the age of muscle cells is a strong predictor of future risk for ALS. Again, based on 50,000 people in the UK Biobank. So we have a blood sample from people when they enter the study healthy between age 40 to 60. We measure their blood proteins with the Olink platform, and we derive signatures of different cell types. We can derive about 40 different cell type signatures based on proteins that are cell type specific, and we can measure in the blood. And so we can estimate how old your skeletal myocytes are, your muscle cells. And if they show an accelerated aging phenotype, you're 3 times more likely to develop ALS in the next 15 years.
So this brings us to the paper where you leveraged the UK Biobank population, 54,000 individuals, and developed organ aging estimates relative to known chronological age, and demonstrated, which you've demonstrated before, that aging is heterogeneous, meaning it changes, it's different across different organs, maybe more heterogeneous in some people over others, depending on age probably as well. But that some of these nodes are dominating outcomes of longevity, health span, likelihood of death. How would you explain that?
Yeah, so what you're bringing up is a very interesting point, that when we build these models, we measure proteins that are derived from specific organs. We have about 11 organs where we have good enough signals, and we can measure proteins that are produced in the liver, but not in other organs, or mostly in the liver. And then if we add 5 or 10 proteins together that are all enriched in the liver and we can measure in the blood, and they change with age, we can build a model that estimates the age of the liver. We can then apply this model to every person and ask how well does our model predict your age? And the deviation from that age, which we call an age gap, is a reflection of faster or slower aging of your liver. So if somebody has an accelerated aging of the liver, they're more likely to have cirrhosis later on, and so forth. So we get these organ-specific diseases. But then when we look at these 50,000 people and we ask, how many people have accelerated organ aging? And then how many people have one accelerated organ age, or so one organ that is aging, 2 or 3 or 4. We find mostly people have only one organ showing accelerated aging, consistent with this concept that Mike Snyder came up with, which he called ageotypes. So it seems like you have one organ that seems to get sort of out of control, and that may be the disease you develop later on. But then we do find people who have multiple organs organs older, and we can predict mortality. So we can then ask which organ is the strongest predictor of future death. And it turns out that an old brain and an old immune system are the strongest risk factors.
And this is because we collected those samples almost 20 years ago. And we know who died. We know who got what diseases. We've got those data in those outcome data from the UK Biobank. And so the strongest predictors of death are brain and the immune system.
Yeah, in this UK Biobank population. And what was even more surprising is if you have young brains and young immune systems, you live actually longer. It's not a dramatic increase in longevity, but it's clearly significant.
And it's young relative to your chronological age. And then you're looking at this across, a normally distributed population, presumably, and looking at, the mean of that. And
so the way you do this is basically, you can imagine like anything, height is always a good example, right? If you look at the population, most people are similar size. But then you have some people are very short and some are very tall. So similar, if we predict the age of brains in 50,000 people, most people's brains will match their actual age. So if you're 50, your brain is predicted to be 50 years old. But then some people have a little bit younger, a little bit older. It's not normally distributed all the time. So you often get a skew towards older and that makes maybe sense because our organs get older all the time. But we really look at the outliers, at the very tails, those with the very youngest and those with the very oldest. And that's usually only 1% to 2% of the population. You can make this cut arbitrarily anywhere you want, but we have decided to really look at this top 1% to 2%. And that's where you find the strongest effects or risk predictions, right? Like we know if you smoke, your risk to get lung cancer increases by, twofold. Here we find if you have an older brain, your risk to develop Alzheimer's increases threefold. And if you compare those with the youngest brains and those with the oldest, there's a 12-fold difference in risk to develop Alzheimer's disease.
Wow.
And what about protective? What about the other side of the curve, those folks that are outliers?
On the younger side? Yeah. So again, what we find is those with young brains, they live longer and have a reduced risk to develop Alzheimer's disease. And in a study that will be published next month, I think, in Nature Medicine, we made similar predictions at the cellular level, as I indicated earlier. So we can build models that estimate the age of a cell in your brain or a cell in your muscle. So we get more resolution, basically, not just saying...
At the cell level, you get the resolution at the cell level, actually.
Yeah.
Yeah, that's amazing. That's amazing.
And so there we find a cell type called astrocytes. So this is a very important cell type in the brain that is involved in glucose transport from the blood to neurons. It's involved in modulating synapses and making them work and actually building them. And we can estimate the cell of this cell type in every person based on protein measurements in the blood. And what we saw is even a more dramatic risk association with Alzheimer's disease, particularly
That's interesting.
in individuals who also have the genetic risk factors called ApoE4. So that genetic factor is a predictor of future Alzheimer's disease, similar to having an old brain. It's about a 3-fold increase. And if you have 2 genetic copies of the A4, it's a 10 to 12-fold increased risk for Alzheimer's disease. But what we find, if you also unlucky and you have very old astrocytes, that increases by a further 3-fold.
Wow.
So we find actually in the UK Biobank that almost 40% of individuals who have the genetic risk factors and old astrocytes, almost 40% will have Alzheimer's disease over the next 15 or 17 years.
Which of them do you think is more predictive? The genetics, proteomics, or combination? Because I would like to highlight here the value of having proteomics data, because so far we're really focusing on genetics data, right? How would you see this moving forward? And like a follow-up question is to the clinical relevance. Would you see the clocks to be like a nice tool to having patient stratification for clinical trials, for clinical diagnostic in the future?
Yes, absolutely.
How would you see this moving forward, the field?
Before we change from the astrocyte story, and we can switch over to that, I just want to talk about the oligodendrocytes the pathways that you saw that were important in brain aging, which may be to some extent a function of which proteins you were able to measure, because it is a targeted assay. Yeah, but these were proteins that weren't neurodegenerative proteins that I typically think of, that show up in, say, Parkinson's disease, for example, when you've got these neurons dying. They were myelin structure. They were the sheath on these. Can you just comment a bit about that? And then I absolutely want to talk about the genetics, the proteomics, bringing these modalities together and that importance.
Yeah, it's super interesting. So when we make these models, the algorithm, first we make the assignment out of the thousands of proteins that are on your platform. We make the assignment, where do they potentially come from, based on gene expression data. So if a gene is only expressed in the brain, then we say, okay, this is a brain protein in the blood. And when we then develop a model that estimates the age of the brain, we know, of course, which proteins are making that signature. And we can ask, what is the biological role of these proteins? What is known about them? And as you just mentioned, the proteins that make up the brain age model are largely involved in a synaptic structure that wraps around synapses and is produced in part by oligodendrocytes, maybe also some other cell types. But it reinforces this notion that maybe synapses are the first part that declines as we get older and that get dysfunctional.
That's a good point.
And that's what we may
There's communication.
Yeah, and that's what we may be picking up long before you get amyloid plaques and tangles in the brain.
Amazing.
This is also consistent with parallel studies that we did in cerebrospinal fluid, where we looked at which proteins are strongest associated with cognitive function, independent of amyloid pathology and tau. And there too, we found synaptic proteins are the strongest predictors, and specifically this NPTX2 and YW that we found are very strong predictors 15 years into the future with a hazard ratio of 15 or so. So a 15-fold increased risk if you had very high levels of that protein.
Amazing.
So to come back, what does that all mean, and how do we integrate this with current knowledge? Genetics is incredibly powerful, right? But what we've also learned is that there's relatively few genes that are really predicting your risk to get a specific disease. ApoE4 is a strong exception. That link to Alzheimer's disease is almost unprecedented in any other disease. So we need something on top of genes, and that's what proteins are, because proteins capture where we live. They integrate many different genes. And that's why I call this the endophenotype. They capture our life experiences and our daily challenges. Now, some of these are very noisy, but the beauty of it is if you measure a lot of proteins, you can pick the stable ones that provide information on disease risk 15 years later. And that's really what we can call the proof in the pudding, right? Because we can say what your risk is in an unknown sample from a person we've never seen before. We know nothing about this person except the concentration.
No lifestyle factors, no clinical labs,
we don't need any other
looking.
knowledge. And that answers your question, Sarantis, right? That means you can potentially translate that to the clinic and provide information to a patient about their future risk. And, while that might not be helpful for some diseases at this point, I think you could change lifestyle. And for some, there are drugs. And we started a company to really take advantage of this and hopefully bring it to the consumer in the coming years. Vero Biosciences, that is trying to harness this information where the consumer can then have their blood measured. We give an indication of which organs show accelerated aging. And then with advice from a clinician, the clinician helps interpret the results and what you can do about it. Because, of course, you don't want to just know that you have an old heart. You would like to know, what should I do about it?
You have to regenerate.
Yeah. And then the beauty is that you can follow up 3 months later and say, okay, did my intervention do something?
Exactly.
And this is really what we're working on now, to prove that these measures are functionally tied to outcomes and a specific organ.
And so what would be needed to leverage organ aging clocks as a surrogate endpoint? And a surrogate endpoint would be, shifting your biological age of an organ or overall biological age would be an endpoint in a clinical trial to demonstrate effectiveness of an intervention, either a therapy or, certainly we do clinical trials on some companies support clinical trials on their vitamins or things like this.
Yeah. So what will be required is to keep validating these tools across the community, and it seems a lot of studies have very similar findings and have shown the value. But there's of course more work to be done. I think the platforms have to be stable, whatever from a technical perspective, and I think they show that they can get the same results with repeated testing. And then what we also need to show is that if you measure these, if you make these estimates in a person, once a month over a year, that they're relatively stable, right? That it doesn't change based on the breakfast you had. So again, we need to figure out what are the signatures that really have this predictive value and are stable. And we're getting there. We have such data set, and it seems they're remarkably stable. But then they need to be tested in the traditional placebo-controlled clinical studies. where you show that they track function, and then you can start using them as a secondary endpoint maybe, and then as a primary endpoint. So it's a stepwise validation in the scientific community, then into clinical trials, and they're also stepwise. But I think we can get there.
In the biomarker world, then, Tony, will you envision a model based on proteomic signatures? Because now I have the impression that we are moving from the epigenetic clocks that initially Steve Horvath has launched with, like the methylation profile, to something more functional to the biology, the proteomics. Then the future it will be bringing these proteomic signatures in a model that you can predict. This is how you see this moving forward.
And we definitely want to talk about Rise, another company that is leading the way in their expertise for building such models.
Yeah, so the idea is that you would be able to make models that predict function and that track with efficacy of drugs and allow you then to use these models as surrogates. And this is what another company that is really trying to provide these services that we started, Rise, is partnering with Olink to provide that service. Not everyone has maybe the tools to develop these models, and Teal has assembled a large database that it uses to train the models and can then provide all these services to really across any type of disease and prediction that you can imagine, and see how valid they are.
And I see this as, hastening us to that state by which we do see these as surrogate endpoints. We do see these as ways to confirm and corroborate interventions or even discount interventions. You talked in Hamilton, first author, and your team talked a bit about ibuprofen, about vitamins, looking at associations. I just want to clarify, we talked about causality earlier. Those associations are helpful. They're one step toward understanding causality, but causality is about finding what are the levers, what are the pathways, what are the cells, what are the triggers that are actually moving us toward disease. And those are the magic that pharma can leverage if they meet the right criteria to build therapies to nudge someone out of a disease state back to a healthy state, right? So the vision here is precision medicine being able to treat the individual based upon what their needs are to bring them safely back into a healthy state and extend their health span.
Absolutely, yeah. So as you said, we have been able in the UK Biobank to look at people's lifestyles, people's use of medications, but this is all retrospective, and some of it is self-reported, so it's noisy. It looks very promising. Yeah. It looks interesting. Like I often mention glucosamine, this in our first study with a different platform on 5,000 individuals, we had a signal on glucosamine use associated with younger organs. We felt this was not good enough to report yet because it was a relatively small number of individuals, 5,000. We saw the exact same signal in 50,000 people with the Olink platform, and we reported it then. But it's retrospective. People report, I use glucosamine, that is associated with some younger organs. So now the next step, as you said, is to do a clinical trial where you have people placebo-controlled, they get the supplement or not, and you do these measurements of the clocks and you say, okay, does it actually track with the functional benefits? Does it track with drug use? And so forth. And we're starting to do that. We have a collaboration with Heike Bischoff-Ferrari, who ran the Do Health trial. This was a 3,000 individual trial with vitamin D, exercise, and omega-3 fatty acids. It was a 4-arm trial, placebo-controlled. They had some functional outcomes, and Steve Horvath showed that epigenetically, some people got younger. And so we're now running with Vero and Thiel, we're running the proteomics to say, okay, is there a signal in the protein clocks? And indeed, we see signals suggesting that, you can pick up functional outcomes, and you get also this agreement between epigenetic and proteomic signatures.
Amazing. So can you tell our audience where might they track or keep an eye on some of the interventions that you might be involved in if there are clinical trial calls for participants? I know Mike Snyder has this often, these QR codes to invite participants that are of a particular demographic. Are there opportunities for people to get involved and support the work that your team is doing?
Yeah, certainly they can sign up on Vero for updates, and we're soon going to launch an IRB-approved beta trial. So people can sign up for that. To analyze data sets, they can, of course, reach out to you and work with Olink and Thiel Rise. And otherwise, stay tuned with the publications that we're working on.
It's exciting. Well, so happy to have you. Thank you so much. We had a little bit of a hiatus in our podcast publishing. We had some months off. This is an exciting episode to be our first one back. So thank you so much for agreeing.
Thank you so much, Tony.
Yeah, thank you. It was fun.
We can stay all day asking you questions.
Exactly.
I mean, we have to stop at the point because they have so many questions that popping up all the time. It's definitely we'll catch up again soon. Definitely we'll catch up.
Yeah.
Because there's so many things we need to know from you.
Thanks so much.
Thank you so much.
Well, that wraps up this episode of Proteomics in Proximity. Huge thanks to our guests and authors of such impactful publications. I also want to thank you for tuning in. Really appreciate you being here. If you enjoyed the content of this episode, please think about sharing it with friends or colleagues you think might be interested in the content. In addition, if you'd be willing to head over to Apple or Spotify or wherever you digest your podcasts and give us a rating and review, this will help others find the podcast when they're searching for proteomics or precision medicine podcasts. And mostly, I want to say we would love to hear from you. So we have a dedicated email address, pip@olink.com. Please reach out. Let us know what you're interested in hearing about, what you care about, and any feedback on the episodes that we have already done so far. This is all about you, and so we're really keen to make sure that we're meeting what you'd like to hear about. Thank you so much, and we'll see you soon.